Hello everyone, my name is Devin Johnson. Today I'll be briefly discussing my research project on the development of a novel 2D tunable hydrogel model to study liver fibrosis. So essentially what liver fibrosis is the progressional stiffness of the liver. This can happen throughout our lives, but it can also be enhanced due to reasons such as what we eat in our diets, lifestyle habits, and also due to genetic reasons. Since 1980, there's been a significant increase in the mortality rate due to liver fibrosis, and this significant health concern has caused the necessity for us to improve the way that we're screening and testing drugs for this treatment of this disease, as well as hopefully one day discover patient-specific treatments. Now, the goal behind my project is to develop a novel modeling platform that can be used to study this disease, as well as show us how these cells are interacting with each other. So the way I went about this was I developed a polyacrylamide hydrogel testing platform in which I created these hydrogels at varying stiffnesses, specifically 8 kilopascal and 34 kilopascal, to replicate that of a healthy and disease-like liver state respectively. In these models, we then seeded human dermal fibroblasts or HDF cells. These cells were then given a control media, vehicle control media, and a 5 nanogram per mil TGF beta media. TGF beta stands for transforming growth factor beta, and this is a protein that induces the activation of these HDF cells. And what we're studying in this platform is to see how do these cells activate and go through transdifferentiation from fibroblasts into myofibroblasts, as myofibroblasts are these cells that form and develop the scar tissue within your liver. By using this model, we can better understand how this disease progresses, as well as how are these cells responding and communicating with one another. Now, for this project, what I did was I had to do the hydrogel fabrication as well as the immunofluorescence staining for imaging and the QRT PCR gene expression quantification. Now, from these trials in immunofluorescence, we stained for collagen 1A as well as alpha SMA. These are two proteins that are commonly related to liver fibrosis. We saw a significant increase with the increase in stiffness. And we also saw the same with the COL1A1 gene as well as the ACTA2 gene. These two were significantly increased with the increase in stiffness. Now, moving forward, I would like to develop a 3D testing platform with the use of organoids. This will allow us to see how multiple cell types interact with each other, as well as see how these cells engage with their ECM varying stiffness environments. And also, these 3D platforms could hopefully one day be used for patient-specific testing. What you could do is take a biopsy of a patient's liver and see how their cells respond to what treatments and what would be most ideal for the patient. Thank you.